Today, I will discuss an important biological phenomenon in human body called carbon dioxide transport and management, which is catalyzed by as well as controlled by some metal enzyme. Next, at first, let's see how carbon dioxide is generated in our body, and then it is transported through blood and get released from our body. That is, it is managed by our body. As we know, after taking meal, the food is stored in our cell in different tissues, and when that food inside the cell get burned and converted to energy, and some carbon dioxide is released. That process is called Krebs cycle, and that carbon dioxide get dissolved in water of blood serum, producing carbonic acid as shown in the equation. Again, we know very well, in the tissue or nearer the tissue, the concentration of hemoglobin, rather deoxyhemoglobin, is very high. Since hemoglobin do not transport or get bind with oxygen at lower partial pressure of oxygen which is present in the cell or tissue, and that deoxyhemoglobin is very good conjugate base to absorb the proton from carbonic acid, and bicarbonate deoxyhemoglobin salt or conjugate acid base pair is formed inside the cell or blood plasma. Now that bicarbonate is transported to the lung through blood plasma in the form of conjugate acid base pair of deoxyhemoglobin. A very simplified cartoon diagram is shown in the left side, where this overall phenomenon is taking place inside the hemoglobin. Again, in the lung, when that bicarbonate adduct is reached and it converted to carbonic acid, and deoxyhemoglobin get converted to oxyhemoglobin, as the concentration or the partial pressure of oxygen is very much high in the lung, and the following reaction takes place. Again, this carbonic acid is immediately get converted to carbon dioxide and water as the following equation. This carbon dioxide get released from the lung as well as our body, and this process may occur spontaneously or some catalyzed present in our body get catalyzed the reaction, and that catalyst is nothing but carbonic anhydrase. And a simplified cartoon diagram is shown right side, where bicarbonate adduct to carbonic acid and carbonic acid to carbon dioxide conversion are shown. Next, as I told in the last slide, the catalyst which catalyzes the reversible hydration of carbon dioxide in our body is carbonic anhydrase. In this slide, a little bit information about carbonic anhydrase are described. This is the cartoon diagram of carbonic anhydrase, where the amino acids as well as the active sites are shown. Carbonic anhydrase was the first discovered zinc metal enzyme around 1940. It is found in animals, plants, and some microorganisms. Another important fact, carbonic anhydrase present in plant do not contain any metal ion in its active site. As we know from our previous discussion, the reversible hydration of carbon dioxide during respiration taking place as shown in the equation, which is catalyzed by carbonic anhydrase. If the reaction occurred without any catalyst and in the body temperature, the rate of the reaction is very slow. The rate constant will be 10 to the power minus 1 per second at 1 atmospheric partial pressure of carbon dioxide. But in presence of human carbonic anhydrase, that means HCA, the same reaction rate get enhanced about 10 to the power 7 times. That means 10 to the power 6 per second will be the rate constant in presence of catalyst, which is the highest known biological rate till now. This Metalloenzyme also function as a catalyst of hydrolysis of ester as well as aldehyde in our body, as shown in the equation. As well as in RBC, the enzyme perform the important role of receiving carbon dioxide from tissue in the active site, active muscles, and releasing it in the lung from our body. Another interesting fact about HCA that it can hydrate around 1 million molecule of carbon dioxide per second at body temperature. Next, now in this slide we will see some active site structure of HCA. This is the active site structure of HCA. A number of closely related form of carbonic anhydrase are found in mammals. Its molecular weight around 30,000 Dalton. More precisely, zinc to iron is present in the center of the enzyme, and it is coordinated by 4 ligands around it, which are 3 nitrogen atoms of histidine, numbered histidine 119, histidine 94 and histidine 96 residues, and the 4th position is occupied by a water molecule as shown in the figure. So overall structure is nothing but a distorted tetrahedral molecule. Again, the molecule is roughly looks like ellipsoidal structure in which zinc to iron lies 12 Armstrong depth cleft inside the enzyme cavity. Again, some other amino acids present near the active site of this enzyme, which play important role during the catalysis of hydration of carbon dioxide in the human body, may function as hydrogen bond formation, proton transfer, etc. In the left side below, the crystal structure of carbonic anhydrase is shown. It is purified, isolated, and single crystal structure obtained by X-ray analysis. Here also we can see the few amino acid residues are present nearer the active site structure and bonded by hydrogen bond formation. Next, in this slide we will discuss another important part, why nature choose or select zinc 2 in the active part of the enzyme Ca, carbonic anhydrase. So what are the importance of zinc? These are the points. Zinc to iron induce tetrahedral coordinate geometry, and we also know the enzyme carbonic anhydrase also adopt tetrahedral geometry. So it is very important in the case of structural analogy. The second point is zinc 2 show very good interactive chemistry and bonding relation with water. It get It is highly soluble and it get strong interaction with water. Since our body contain almost 70% of water, so this enzymatic function can take place very easily in presence of zinc 2. And the third point is zinc 2 is highly stable and it is very effective Lewis acid. Since it contains vacant 4s and it's higher and also higher orbitals, where it can accept the electron pairs from the ligands, forming coding bond. It is very important for its catalytic activity and show metalloenzyme activity. And the last one, in maximum metalloenzyme, zinc also perform as stabilizing structure as well as structural atom to perform the enzymatic activity. So these are the main features for which nature select zinc 2 during the formation of metalloenzyme Ca. Now zinc 2 iron is very essential for our life also. Deficiency of zinc in our body, a large number of disease or disorder can be observed. They are like rendered growth, anemia, loss of appetite, taste sensitivity, lots of metabolic disorders, etc., etc. Now, to interesting fact that for an adult human, about 15 to 20 milligram of zinc is required per day, and the source of zinc are meat, egg, fish, beans and nuts. Next, catalytic action of carbonic anhydrase. This is the most important part of this lecture. This is the first step of catalytic process where the active site of the enzyme is shown, which is nothing but the distorted tetrahedral one, and water molecule is attached in the fourth coordination site. Now the 3-histidine molecule present below the structure. Since nitrogen molecules are very much electronegative, it polarizes the bonded electron, and as a result ZnO bond as well as OH bond get polarized and H plus ion get released to produce the active site structure as OH form shown in the catalytic cycle. This is the first step of the catalysis, and this step is very much important step for the catalysis. Without this polarization, catalytic activity is not started in the case of carbonic anhydrase. Now the second step, when this active site or vacant active site is generated, the more nucleophilic OH group of catalyst attack to the electrophilic carbon center of the carbon dioxide, which is generated inside the cell, and a rearrangement is taking place like this. And this is the third step of this reaction is very much important, and it is a rate determining step and very important step of the enzymatic action of Ca, where the OH group holding the CO2 minus group attached with the zinc orient itself in such a way that the above attack from CO minus to zinc could take place, as well as a very hindered, unstable 4-member ring is generated, which is the key step of the enzymatic cycle as shown here. Now the last step, after the conformational changes of this 4-member ring and followed by the nucleophilic attack to the zinc center, replacing the or releasing the HCO3 minus from the catalyst, and the initial carbonic anhydrase metal enzyme is returned back and complete the catalytic cycle. And this regenerated catalyst is ready to perform another cycle, another catalytic cycle, to convert carbon dioxide to bicarbonate ion. This is the brief procedure of conversion of or hydration of carbon dioxide to bicarbonate in presence of Ca or HCA. Next, this is the last part of my lecture, that we have seen that zinc 2 is very much important for our body, and it is the main part of the catalyst Ca, and it perform important catalytic cycle during the conversion or reversible conversion of carbon dioxide to carbonate or carbonic acid. Now, what is the drawback of zinc? How we can understand the catalytic process or the mechanism of catalysis by which carbon dioxide get hydrated? These 4 steps, how we will produce the mechanism? The only way is spectroscopical analysis or structural analysis. But since zinc 2 ion is present in the center of the catalyst, as we know, zinc 2 is detained system and all electrons are paired up, so maximum zinc compounds or enzymes are colorless. Hence, they show very poor photophysical and photochemical properties. Hence, no absorption, emission, spectroscopical analysis are applicable for this enzymatic process. Again, since zinc 2 is diamagnetic, hence EPR and MOSBO are silent. So it is very much difficult to identify this type of enzyme and its enzymatic activity in vivo. So what is the solution of this problem? Only solution are the molecular modeling in vitro. If we change or transfer or replace the zinc 2 from the active site of metal enzyme Ca by cobalt 2 ion D7 or nickel 2 ion D8 or copper 2 ion D9 ions, which are highly colored and spectroscopically highly active, and we get data from their compounds in different stages of the catalyst: absorption, emission, CD, the magnetic CD, and as well as the X-ray analysis if we could crystallize the compound. So by this molecular modeling, we can check or we can study the mechanism of the catalytic activity in vitro and confirm the catalytic process performed by Ca. So that's all about today's discussion on management and transport of CO2 in human body in presence of some zinc 2 metal enzyme. Thank you.